Okay, so we're going to keep talking about the factors that cause populations to evolve. So remember in the first lecture, we're talking about to measure evolution, you have to track genetic frequency within a population, not an individual. I want to stress that, and I'll keep stressing it. Individuals do not evolve. They can adapt, but they do not evolve. Evolution is the change in the gene frequencies of the population over time. So we're looking at the genes, the alleles, big A's, big B's, little b's, etc. And are those ratios changing from one generation to the next, to the next, to the next? So we talked about the first two factors, mutation being one, and then this gene flow being the second. So now we want to get into our third factor. The third factor that can cause populations to change. And this is what we call non-random, or you may see it called assortative mating. What this is sexual selection when organisms go to mate. Now this does not apply across all species, obviously. You got plenty of species that do not choose. Plants, they don't choose who they mate with. The bee does that. But look at a lot of animals. Lions, dogs, bears, fish, etc. There's a mating process. There's a mating dance. There's a behavioral thing going on. There's a ritual. There's a competition, there's fighting for the opportunity, etc., and there's choice. Typically, the most adaptable are the ones that get to mate. So you think about sexual selection or selective breeding, in general, likes tend to breed with likes. So for those of you who watch the dating shows like The Bachelor, The Bachelorette, think about all the dating websites, eHarmony and all those kind of things. What you're doing is being selective about who you mate with, who you're going to reproduce with. You're looking for certain traits that you desire, that are specific to your interests and your likes. Likes tend to breed with likes. So our bachelorette here, going out for the evening, chooses between all these different guys. You can get a good picture of who she's going to mate with. All right, so when you have this assortative mating, certain genes are more likely to be passed on. The ones who can't mate don't get to pass on their genes. So again, this doesn't apply across all species. You know, we have plenty of species, again, plants, they're not selective. They just simply, pollen gets released and hopefully it gets transferred to the egg of the same species. Not really a lot of selection there. So they're not using this as a factor for evolution, but mostly it's the animal world. When you get into non-random mating, that can drive evolution. So if if the females in a population all have a certain preference for males with a certain hair color or a certain behavior, those are the individuals that get to mate. They pass on the genetics to their offspring. Their offspring probably have that same behavior. Those female offspring probably have the same preference, and then that trait continues to get passed on generation after generation while the other traits decrease in frequency. All right, so non-random mating is our third variable or third factor. Now the fourth factor is called genetic drift. This is a change in gene frequency due to chance. Gene. No. All right. Population size is incredibly important here. All right, so let me scoot this down so I can put an example here. All right, so we got two wolves. They killed a deer. That happens all the time. Now let's say we have a deer population, or populations. On the left-hand side, we're going to have population A, and there are 10 deer. On the right hand side we have population B, and there are 100 deer. Now the genes we're looking at will be big H and little h. So on the left hand side, let's say big H is 90% of the population and little h is 10% of the population. So assumably, and we're just going to make up some examples here, big H is dominant. Let's say 9 of the deer have the big H. Little h is recessive. Let's say to be fully expressing, you have 2 little h's. Only 1 deer is going to express a recessive trait in that population, a recessive variation. Now with the bigger population, we're going to still work with the same frequencies. H, 90%. That means 90 deer have the big H's. The little h's are still 10%, and this means 10 of the deer in the bigger population have the recessive allele. So if the wolves are out hunting and they happen to go after the smaller population of deer and they kill the one recessive deer, that deer's dead, all of a sudden your gene frequency changes instantly. It's now 100% dominant allele. 100% expressing the dominant variation. If that happens over here, the wolves kill 1 deer. You have 9 deer that are recessive. You have 90 deer that are dominant expression. You don't see as big of a change. Maybe you're at 91 versus 8% or 9% of the recessive in that bigger population. So drift can happen at any time. It happens all the time with populations. Now there are 2 types of drift. These are subcategories that go underneath genetic drift. Bottleneck is one, so I'm going to put A, so we know this is a subcategory. And in the bottleneck effect, a large population crashes down to a smaller population. When this happens, you lose some of your genes. Alright, so the best examples, I'll show you the next slide, it's got marbles. Alright, so we started with blue and green and red and yellow marbles in those ratios. Big population, lots of individuals running around, lots of alleles. And for whatever reason, the population crashed. Now that could be disease, it could be overhunting, it could be habitat loss, it could be 1,000 different reasons. But the population size got reduced. And when it got reduced, all of the yellow alleles, the individuals who had that gene, died. We see the green alleles being more frequent in the smaller population. For whatever reason, those individuals survived the crash better than the yellow alleles. They survived it a little bit better than the green and a little bit better than the red. I'm sorry, the blue and the red. Both the blue and the red frequencies dropped, green frequencies went up. That's a big issue, and especially since you've lost yellow alleles. Maybe that was yellow feathers in the birds or whatever this is. You've lost it. Now those alleles are gone. That's a problem. Your population is now very different. That could happen in a very short period of time if environmental factors trigger a population crash. So the cheetah is a good example of a bottleneck. It's estimated about 10,000 years ago the cheetahs crashed in population size all the way down to 6 to 8 cats. Why? We're not sure. Could have been overhunting, could have been habitat, disease. Most likely it was a combination of a lot of factors. But 6 to 8 individuals. They've rebuilt their population from those 6 to 8 individuals, which means they're inbred beyond belief. They're genetically related to each other, very, very closely related genetically because of this bottleneck effect. So consider when populations crash, the remaining individuals are the base of the gene pool they're the ones that reproduce and start rebuilding the population. The population grows, and individuals mate with similar individuals that are potentially distant relatives, and it keeps the gene pool fairly contained. That often leads to genetic problems, and the cheetahs have some genetic problems. The majority of the males in the wild are, because of the inbreeding, are sterile. They have a very difficult time reproducing naturally in the wild because of this inbreeding issue. All right, so that is one type of what we call the genetic drift factor. And then the other one is known as the founder effect. Now, the founder effect, this is going to be your B for the genetic drift. The founder effect is when your population is reduced, but you don't crash the whole population. You just isolate a small group. They get removed, isolated, separated, whatever the case may be. So, our example with our mice. Original population, you had 5 black, 5 white mice. You have a 50-50 ratio. The dam breaks, the river forms, the mouse population is now split in half, and they're isolated. They cannot get back together to reproduce. So the population on the right is now 80% black, 20% white. The population on the left is 80% white, 20% black. That's going to influence those genetics. And over time, those mice will continue to reproduce, and those gene pools will start to go in different directions. So they cannot get back together because of that river that has created 2 separate populations. In humans, we have a similar example with the Amish. The Amish, for the most part, are isolated reproductively from the rest of the human population. Odds are nobody listening to this lecture or watching it will ever reproduce with an Amish individual. So the Amish, for the most part, are closed genetically to the rest of society. So because of this and how they've remained closed for hundreds of years, they have a greater frequency of dwarfism and this trait called polydactylism. It means you have 6 digits than the general human population. Now, they're still people, they're still humans, but they just have different frequencies in their gene pool. Now, right now, we're not going to say, oh, they're completely different. Not at all. But let's jump forward a couple thousand years. What we might find, if the Amish remain closed and isolated, we might find that if you went to an Amish community, you might find the majority of the people are dwarfs. Maybe that is a beneficial trait. When you're shorter, when you're smaller as a dwarf, you require less food, less energy, etc. So we don't know what the future will hold for the Amish with the founder effect. But if you remain in a closed gene pool, it allows your population to evolve differently than the rest of the general population because you're controlling the genes by remaining somewhat of a closed gene pool. Consider dogs. If you have a purebred dog and it's a closed gene pool, like, oh, I have a purebred Malamute or purebred Shepherd or purebred Husky or purebred Cocker Spaniel, that dog is in a closed gene pool, meaning it is only breeding, or its parents and grandparents, etc., were only bred to other purebred dogs. And in dog breeding, there's a lot of bad genetics. They have intentionally created the founder effect by saying, we only want specific dogs to be allowed to reproduce to maintain this breed. They've closed the gene pool. There's a lot of inbreeding to create purebred dogs, to get those desired traits. That leads to a lot of genetic issues, health issues associated with purebred dogs. So, founder effect with the Amish, same concerns. Are there going to be long-term genetic health issues associated with those closed populations? All right, so those are the 4. We still have one more factor that causes frequencies to change. We'll take a look at that in the next lecture, and then we'll wrap up everything in regards to looking at how do populations evolve.